Hi class, this is lecture 13, the chromosomal basis of inheritance. So we covered genetics and how Mendel came up with some foundational principles about genetics and what we know. And we also covered how some things may differ from Mendelian genetics or non-Mendelian genetics. And so this lecture is going to focus entirely on chromosomes. So what is the relationship between genes, which we talked about, and chromosomes? So we're going to go one by one. So genes are located on chromosomes. They are segments of DNA that code for something. And so they are located on chromosomes. And so if we look at the top of this figure, either on the left or on the right, these are two separate, parental cells. And so each parental cell is diploid, so it has two chromosomes of this chromosome. So for example, chromosome 1. And in chromosome 1, there are these recipes for things. And then on each of these homologous chromosomes, there are alleles, which are alternative forms of those same recipes, right? So like flower color, either purple or white flower color. Tall or short plants, for example. So yeah, remember genes, and then alleles are different versions of genes, and so genes are located on chromosomes. And then when chromosomes duplicate before cell division, that's how you get the sister chromatids of each homologous chromosome. So before cell division, the chromosomes duplicate so that there is enough to get divided up. During meiosis 1, so those homologous chromosomes get separated from each other, and so those alleles get separated from each other. And in meiosis 2, the sister chromatids get separated. So at the end, you have 4 different gametes that have either this allele or maybe a combination of this and other alleles in each gamete. So the gametes are haploid and only have 1 copy of each, because they come together in fertilization and make a zygote, which is diploid again. So it has 2 copies again of the homologous chromosomes. So each chromosome has 1 version of a gene, so 1 allele, and then when they come together, offspring inherit 1 allele from each parent. So this is combining the past 3 lectures, basically. Meiosis, genetics, and then here we kind of focus more on the chromosome. So all of these things do come together. Even though they were separate lectures, they're all tied. So Mendel had proposed something gets passed on, and back in 1860, he didn't have like a real concept for what it was, so he just called it hereditary units. And so they were just theoretical at that time. And so soon biologists saw these parallels between chromosome behavior and the behavior of these hereditary units, or proposed like factors. And so they kind of made this connection of maybe that's what it is. It's the chromosomes getting passed on. And so to kind of continue this story of how scientists came up with all this, so around 1902, Sutton and Boveri, as well as others, they independently noted that these parallels, and so they came up with the chromosome theory of inheritance to try to make sense of all this. So the chromosome theory of inheritance is basically putting into words like it's chromosomes that we inherit. Those are the actual things, the hereditary units. So the first solid evidence associating a specific gene with a specific chromosome came in the early 1900s from the work of Thomas Hunt Morgan. And so we're going to talk a lot about his experiments and how they showed, okay, something is on these chromosomes and it's the genes. So his early experiments provided convincing evidence that the chromosomes are the location of Mendel's heritable factors. So what Thomas Hunt Morgan studied was the fruit fly. So you might notice them at home, the really tiny, annoying fruit flies. But this is actually a big model organism in science. We learned a lot about genetics from the fruit fly. And so their actual scientific name is Drosophila melanogaster. And it's a common species of fruit fly. And so it's super common, so that's part of why he was able to use it as like a model organism. Kind of how Mendel, like, oh, it's easy for me to get these pea plants. There's a lot of variety. So several characteristics are what made fruit flies a convenient organism for him and for still, genetic studies. We use the fruit fly a lot. And so it's because they produce many offspring. So again, it's really nice to be able to study genetics and inheritance when something has many offspring versus humans that have just one at a time. That would take forever to study genetics, right? Or like elephants. So it's nice because fruit flies have many offspring, so you could see these patterns more quickly. A generation can be bred every 2 weeks, so also very fast. Again, it's not like, a long gestation period of like 9 months. You can have a new generation in 2 weeks. And it's really convenient that they only have 4 pairs of chromosomes. So it's very easy to kind of track and see these things get passed along versus if there was like 46 chromosomes. That's very complicated, harder to kind of keep track of everything. So yeah, the fact that they only had 4 pairs of chromosomes was also pretty convenient. And so Morgan noted phenotypes. So remember, phenotypes are what something looks like, what gets expressed, what shows in a photograph. So he took note of phenotypes that were like normal, or pretty much common in the fly populations. So that way he can have kind of like a baseline. So he noted what is typically their most common color, or how do their wings normally look? What about their eyes? What color are they normally? So this is called wild type in genetics. What you see as the most common in the population is what is called the wild type. So basically like normal. And then you can also start to notice like small differences. So like if normally they have red eyes, it's a little bit new if they have white eyes, So those are wild type phenotypes that he took note of. And so traits that are alternative to the wild type are called mutant phenotypes. So yeah, for red eyes, that's wild type. That's kind of normal. That's most common. And so the first mutant that he noticed was a fly with white eyes instead of the red eyes. And so that is called a mutant phenotype because it's not the norm. It's not what's most common in the wild. Usually it comes from a mutation, so that's why they're called mutant phenotypes. So here's an example. The wild type, again, for eye color is red eyes. We don't really pay attention to the fruit flies at home, but if you were to look, most likely they're going to be red. It's most common. And white eyes are called the mutant phenotype because they're just different. So he studied these fruit flies for years and years, and for a while he wasn't getting anything different. They were mostly the wild type phenotype, and then he found some that were had white eyes. I was like, oh, okay, interesting. Finally, something. So in one experiment, he was curious about what the red versus white alleles came down to, basically. So in one experiment, he mated male flies with white eyes, and then female flies with red eyes to see what would happen. What is the pattern? And so that, remember, is the P generation for parents. The next generation, the F1 generation, all had red eyes. And so again, kind of like Mendel, he was like, okay, it's kind of like the purple flower color. In the F1, they all show purple. So let's mate them again to see what would happen in the next generation. And so when he mated the F1 to another one, the F2 generation showed a 3-to-1 ratio of like red eyes to white eyes, but only the males had the white eyes. And so this was different than like the purple and white flowers, right? It was like purple 3-to-1. But because we're also observing their sex, he also noticed, well, it's only males that had white eyes. It's not the females that ever have white eyes, really. So this prompted, more interest. So Morgan reasoned that the white eye mutant allele must be on something that can only end up being expressed in males for some reason, right? And so he reasoned that the white-eyed mutant allele must be on the X chromosome because female flies have 2 X chromosomes. And then male flies only have 1 X and 1 Y. So if the X in a particular male is the mutant white-eyed allele, then again, that's the only way it could be expressed because it can't get covered up by the other wild-type allele. So a female fruit fly can carry the mutant allele for the white eyes, but if there's going to be a red eye as well, it's dominant basically, so it covers it up. So it can only be expressed, the white eyes, just like the white flower, if there's only the recessive present. And so that's easier in males because there's only 1 X. And so if there's the white-eyed allele, it's going to get expressed. And so all of these findings supported that chromosome theory of inheritance. It's the chromosomes that carry the genes, and that's how they get passed on. So here's a visual example because it can be kind of hard to like abstract. So this parental generation, the P generation, he had a female. So this is the symbol for female. And I think they said it's because it resembles like a mirror. So if you ever have a question about that, if it's like standing up and it looks like a mirror, then that's supposed to be the symbol for female. So a female fruit fly with red eyes, wild type, and then a male fruit fly with white eyes. And so this is male. I think it's because it's like a bow and arrow, whatever. So when he mated them, the F1 generation all had red eyes. So again, he was like, yeah, it's kind of like the purple flower color. All of the F1s were purple. Let's cross this one with another one because fruit flies cannot self-fertilize. So yeah, you cross them. And so in the F2 generation, what he saw was the 3-to-1, but that the white eyes only ever showed in males. It never shows in the females, basically. And so he thought, okay, that's interesting. Like something must be up with that because this 3-to-1 ratio is the 1 is always going to be in a male. So when you show the chromosomes like this helps make a little bit more sense. So these are representing the X chromosomes, this shape. And then the color is just representing, it's coming from the female fly. And then so again on this side, here's the shape for the X chromosome, and then here's a weird different shape for the Y chromosome. And then they're just blue to show it's from the male side. So if we were to kind of do the Punnett square, and this is basically what would happen through meiosis. So the eggs basically could only have this or this, the wild type allele on the X chromosome. So wild type is shown by like a W and then a plus. So wild type in general for any other trait is going to be like a plus, and then the letter is just showing the allele, kind of like the big P, little p. So a capital W, and then the plus means it's a wild type. And then here is recessive, little w, and it's mutant, so it doesn't have the plus. Okay, so here we see the meiosis. Either way, basically the gametes possible here are going to be this. So they only showed one row in this Punnett square. So that's like a quick time saver. So it's either going to be this or this, which is the same thing. So here they're representing these are the possible gametes from these. These are the possible gametes from this guy because they can either get a copy of the X shown by this shape or the copy of the Y shown by this shape. Because again, gametes are haploid. So it's kind of like 50-50 what this gamete's can end up with. And then in this fertilization, because again, Punnett squares are representing like fertilization and the zygote in that potential combination. So here is a red-eyed female because this gamete happened to get the X chromosome from each one. And so there's 2 X chromosomes, so this fly is female. She got a wild type allele. So the plus is wild type, and that's dominant. And so again, if there's even one copy of that, it's going to overshadow the other one. So she has red eyes. And then in the other case, if it happens to be a sperm with a Y chromosome meeting with an egg with the X chromosome, then this male has 1 X and 1 Y chromosome. And this gene is only carried on the X chromosome. So here on this X, it's wild type. And so these males happen to have red eyes because there's no white chromosome, right? Or white allele. So again, if you take one of these and cross it with one of these, then what happens in the F2 generation? And so this is those combinations. So again, setting up a Punnett square, you're looking at what are the different possible gametes from this diploid organism. So the gamete could either end up with the red X or this blue one. And again, the plus indicates wild type, so red eyes. And then no plus means the mutant allele. So in this combination, it's red eyes because they have both copies wild type. So for sure, red eyes. This one has 1 copy wild type and 1 copy of the mutant allele. But again, if this is present, it's going to be expressed. So red eyes in this female, because again, there's 2 X's. And then if these guys happen to inherit a Y chromosome, they're going to have the Y chromosome and be male. If they inherit this chromosome with the plus, again, that's wild type, so it's going to be red eyes. And then in this case, there's the Y chromosome, but they actually get the mutant allele. And so because there's no wild type to cover it up, it's going to express that wild white eyes allele in this male. And that's the only combination that's going to show that. So hopefully that helps make sense a little bit more. So now that we can put, okay, it's chromosomes that carry these genes, we can also apply this to make a little bit more sense of Mendel's laws. So Mendel, came up with these laws of like segregation and law of independent assortment without even knowing what chromosomes were. And now we can like depict them with chromosomes. So the law of segregation, he said just the 2 different alleles for one gene are going to separate. And so if we show that here, it makes sense because the chromosomes physically separate. So the 2 different alleles for a gene get separated. Bada bing, bada boom. Sorry. Okay. So those alleles physically get separated from each other because they get divided up during meiosis. And then I think what's even more impressive is that he came up with the law of independent assortment. And so he said that the alleles of genes on different chromosomes that are non-homologous assort independently from each other. So it's not always that dominants go together or recessives always go together, like they assort independently of each other. And so now we can show that with chromosomes. So, for example, the yellow seeds and round seeds. So yellow being Y and then round being R. So these two totally different traits that he followed. And in this case, he got lucky because they were on different chromosomes. So as we go through meiosis, you can see how they do assort independently. Like it could be the case that the dominants end up on this side. And so there are gonna be some that are round and yellow. But it's also still the case that you're gonna have some of the other combinations because there's gonna be some that are green and smooth and round. But then you also see the other ones because of this. Like they could assort independently of each other so that the dominant for this one is on this side. And then the dominant for this one is on this side. And so in this random, independent assortment, that's how you get this combination and this combination. And then that's how these happen. So yeah, it was pretty impressive that like Mendel did all of this work without even knowing what it was that carried all these things. But now it makes sense with our knowledge and hindsight that it's chromosomes. But again, not everything is so kind of clean cut as what Mendel was studying, because there are some more complex cases where there are things that are linked. So what Morgan discovered was a trait that correlated with the sex of flies. And so that was key to the development of the chromosome theory of inheritance and what we now know are sex-linked genes. So humans and many other mammals have two types of sex chromosomes. So remember, there are autosomes that code for basically everything else, and then the last set are the sex chromosomes. And so there are two types of sex chromosomes. There is a much larger, like literally physically larger X chromosome, and then a much smaller Y chromosome. It's so cute. And then so these are homologous chromosomes. Sorry, sister chromatids. And so yeah, this is much larger X and then the smaller Y. And so a person with two X chromosomes usually develops anatomy that we associate with the female sex, like biological sex. And then male properties are associated with inheritance of one X and one Y. And that's not the only system, like the X and Y system, just because it's also present, in humans, many other mammals, and flies. It's not the only one. There are different chromosomal systems of sex determination. So humans, yeah, we follow the XY system. So there's two X's or an X and a Y. But there are other ones as well. There is the XO system, or X0 basically. So grasshoppers, for example, or like the fruit flies that I used to work with. Not fruit flies, the worms. So you either have two X's and are female, or just one X. There's like nothing else. There's no Y. So that's why it's X0 or XO. And that means male. But there's also the ZY system. So you can have a Z and ZW, sorry. Z and a W and be female, or a Z and a Z and be male. Or in bees, for example, there's just the haplodiploid system. So you're either diploid in female or haploid in male. So there's a lot of different chromosomal systems of sex determination, basically. But we're mainly going to focus here on the XY. And so there are short segments at the ends of the Y chromosome that are homologous with the X. So that's how they can kind of behave like homologs during meiosis in males and still get passed, evenly. And in mammals, again, like us, a gene on the Y chromosome called the SRY gene, and that stands for sex determining region on the Y, is what's actually responsible for development of the testes. And so a gene that is located on either of those sex chromosomes is called a sex-linked gene because it's literally physically linked to those sex chromosomes. So genes on the Y chromosome are called Y-linked genes, like this one, the SRY. But there's not that many of them because the Y chromosome is so small. So on the Y, there's only 78 genes, and they code for about 25 proteins that have been identified on the human Y chromosome. And then the X chromosome is so much larger that it contains about 1,100 genes. So genes on the X chromosome are called X-linked genes. And there's a lot more of them. So there's only 78 genes here versus 1,100. So on the X chromosomes, there are many genes, in general, but there's also genes for many characters unrelated to sex. So it's not just like this is all it takes to be a female. It's also other stuff that's unrelated to sex. But many of the Y-linked genes are related to sex determination. Again, there's just like not that many, so comparatively. So X-linked genes follow a specific pattern of inheritance, kind of as we visually showed. If there is a sex-linked gene on the X chromosome, then it depends on if you end up with just one X, because there's a Y, or with two X. So for a recessive X-linked trait to be expressed, a female needs two copies of that allele. They have to be homozygous to express and show a recessive allele. A male only needs one copy of the allele for an X-linked recessive trait to be expressed. And in this case, because they only have one, it's hemizygous, because hemi is kind of like half. So a male only needs one copy of the allele. Because there's not two X's, they can't be heterozygous, right? So they are hemizygous because they only have one X. So they only need that one X to express a recessive X-linked trait. So this means that anything that is X-linked and recessive is going to be much more common in males than in females. So for example, color blindness. It's a lot more common for color blindness to be expressed in males because they only have one X chromosome. So if that one X chromosome has a recessive X-linked trait, they're going to express it. So they're more likely to be colorblind. Females can be colorblind, but it's just not as common because they happen to need to inherit two copies of that recessive X-linked trait to be able to express that and be colorblind. So yeah, if a female is colorblind, you'll have to look at the parents, like they both have to be, or the dad for sure has to be. So here is an example of a recessive case. So here we just have N for anything, but let's imagine it's color blindness. So if it's recessive, it's a, lowercase n, and dominant, not colorblind, is a capital N. And if it's sex-linked, then yeah, you put an X if it's linked to the X chromosome. And then the letter for the allele is like a superscript. So like an exponent, but it's called a superscript. So if it's X linked, then you put it on the X, and then you put what the allele is, capital for dominant or lowercase for recessive. So that's how you track stuff. That takes a little bit of practice to get used to like thinking about. But that's how you track sex-linked traits. So if a male has this combination of sex chromosomes, so a recessive X-linked trait, and then they have one X and then one Y. So that's what they could pass on. Their gametes can either have the X with a recessive or Y. And then this female organism, whatever it is, again, if we're thinking about color blindness, so she can have either gametes inherit this X with the dominant or this X with the dominant allele. So in this case, they happen to inherit, in this particular combination, this offspring could have, they could be heterozygous. So they have, this is female because there's two X. And then because they have one dominant copy, they're not going to express that recessive allele, but they are carrying it. They're carriers. And then the same for this one. They are technically heterozygous, but they're going to be phenotypically like normal because they have the normal allele or dominant allele. And then in this generation, the males are going to be phenotypically normal, not colorblind, for example, because they inherited this X chromosome from the mom. And so if one of these happens to meet somebody else, and this is there, so if this female offspring happens to meet somebody else, and then this is this guy's genotype, so if they have kids, then this is the possible outcome. So it could be that, this is like again, not colorblind male. And so in these different combinations, it's more likely that all of the female offspring are going to be, again, heterozygous, so phenotypically normal, not colorblind. But in the sons in this generation, there's a 1 in 4 chance that they will be colorblind and get the recessive allele from the X chromosome of the mom because she was a carrier. So even if the male, the dad, doesn't have it, if the mom is a carrier, there's a 1 in 4 chance that one of the sons or one of the kids will be a son and have the color blindness or the X-linked recessive trait. And so that is the case if like one of the daughters meets a male that happens to have this genotype, is not colorblind. And then if this is the one of the daughters is a carrier of the X-linked recessive trait, so again, example colorblindness, and she happens to meet somebody who is colorblind, then he can carry on this X-linked recessive trait, and so there's a higher likelihood of their kids being colorblind. So in this case, there's a 50-50 chance, basically, 1 out of 2 chance that they will be colorblind. And it's a 1 out of 4 chance that it'll be male colorblind or female colorblind. But again, every kid is going to have that 1 in 4 chance. So in this combination, if a female carrier happens to mate with a male who has this recessive trait and is X-linked, then these are the different outcomes that are possible. So it is a lot more common if you take this odd and this odd, right? For males to be colorblind or any other X-linked recessive trait. It's a little bit like smaller chances of a female having the trait. But it's all, just on odds, basically. So, for example, some disorders that are caused by recessive alleles on the X chromosome in humans, like I mentioned, colorblindness is mostly X-linked. There are some exceptions. And then there are other disorders like Duchenne's muscular dystrophy or hemophilia, where people are more likely to bleed because they have an absence or deficiency in blood clotting factors. So if somebody gets a cut, it's not going to stop bleeding as easily. So those are some other examples that are X-linked. And in mammalian females, we get 2 X chromosomes, but one of the 2 X chromosomes in each cell is randomly inactivated during embryonic development. So only one really gets expressed, the other one gets inactivated. So the inactive X condenses into what is called a Barr body. And this is, again, randomly in development. So in embryonic development, some of the cells will randomly turn off one of the X's and other cells might turn off the other one. So it is pretty random. So if a female is heterozygous for a particular gene that is located on the X chromosome, technically you're going to be a mosaic for that character. So some of the cells might have one copy of the X and other cells might have a different copy of the X, or expressing a different copy. And they're not destroyed or anything. It's kind of just silenced a little bit. So inactivation of an X chromosome is involved with modification of the DNA and proteins. So DNA we think of as just loose in DNA, but DNA is wrapped around proteins called histones. So this helps organize and condense it. So a part of the chromosome contains several genes involved in the inactivation process. And one of the genes there becomes active only on the chromosome that will be inactivated. So say that there's 2 X chromosomes. This one's going to be inactivated, so the gene here that's involved with inactivating itself will be activated, if that makes sense. And so that gene is called XIST, so X inactive specific transcript. So that's the gene that's saying hey, we're going to get inactivated for a little bit. And so then it makes itself get inactivated. So for example, this is super interesting, and cat genetics and pet genetics is super interesting. But so an example of the X inactivation being random in different parts of an organism is the tortoiseshell cat. So in a female cat, that's a tortoiseshell. That's because it might be a mix. So if one chromosome has the allele for orange fur, the other chromosome has the allele for black fur. So this female cat is heterozygous for the orange or black fur color. One chromosome has orange, one chromosome has black. And so during development, a few of the cell divisions end up turning off one gene and expressing the other one. So in this case, it's inactivating the gene that had the orange fur and only expressing the one that had the black fur in some cells. And then in other cells, it's the opposite. So this is the active X with the orange fur, and then the inactivated one was the one with the black fur. So that's how they have different cells expressing different coat colors in one organism. And that's not the case for other kinds. It's just in tortoiseshell, so not calico. It's just tortoiseshell. They are female, and you see this different mosaic of expression because of the X inactivation in different cells.